Okay, we're going to talk about fatty acid catabolism, how to break down fatty acids to generate energy, and how to really connect the metabolism of these fats to things we know about already, like reduced cofactors and the Krebs cycle. So we've already talked about how we generally don't have just free fatty acids floating around in the bloodstream. We have, you know, tags, triacylglycerols. Now, those triacylglycerols can be broken down to fatty acids by the action of lipases and the addition of water as part of that reaction. Now, these fatty acids are generally in the cytoplasm of a cell. And, as they cross the plasma membrane where the lipases are and are broken or released as fatty acids, these fatty acids are in the cytoplasm. But in order to break down fully these fatty acids, we have to get them to the mitochondria. And we have to get them in the form of what's called an acyl-CoA, which I'll show you some structures of in a minute. So we're going to have to go through some work in order to get these fatty acids in the mitochondria so we can break them down. And so we're going to activate them. Now, the first step of activation uses an ATP molecule and couples that fatty acid to the AMP. So when we think about accounting, we are just using one ATP, but we're going to count this as if it was two ATPs because we've used two of the high energy phosphate bonds in this adenosine triphosphate to couple the adenosine monophosphate to the fatty acid. So we've lost two high energy bonds. Really, to restore this AMP back to the ATP, we'd have to use two ATPs. So we're using one ATP, but we're going to count it as two because it's two high energy phosphate bonds. Now, once we've coupled that fatty acid to the AMP, we can couple it to our coenzyme A. And this is what we call our acyl-CoA. It's not necessarily an acetyl-CoA. It could be a much longer carbon chain than just two carbons, so we call it an acyl-CoA. We're still thinking this is cytoplasmic. We haven't changed anything. We've just coupled it to the CoA. So now we're going to have to shuttle this across the mitochondrial membrane. And there is a shuttle that I will discuss called the carnitine shuttle, which takes that carbon chain and pushes it across the mitochondrial membrane where we can break it down. Okay. Now, once that fatty acid is in the mitochondria, we can undergo a process called beta oxidation. And beta oxidation has products that we recognize. We're really going to chop this acyl group, this carbon chain, into two carbon pieces. And so one of the products of beta oxidation is acetyl-CoA. We know what to do with that, right? That can go right into the Krebs cycle to generate energy. Another product of beta oxidation is our FADH2 and NADH. Those aren't surprising. Since we're oxidizing our carbon chain in beta oxidation, we can reduce other things like these cofactors. Now, these are in the mitochondria also. We know what to do with them. These can be used to generate our protein or a proton gradient that can drive ATP synthesis. And so even though it's going to cost us a little bit of ATP to activate this fatty acid in the first place. Once we get it coupled to our coenzyme and into the mitochondria through a shuttle, then beta oxidation is really going to give us a lot of energy, right? Acetyl-CoAs can be burned through the Krebs cycle to give us all the products we're used to, carbon dioxide, FADH2, NADH, GTP. But also, just in the process of breaking it down and oxidizing it, we're going to generate some separate cofactors, which can be used to generate ATP through a proton gradient. So there's a couple things in here we have to go into more detail about to understand catabolism. One is the carnitine shuttle. How does that work? And the other, we're going to look at the reactions of beta oxidation to see how we're going to turn this longer carbon chain into acetyl-CoAs and generate those reduced cofactors.